3D intestinal organoid culture limits the study of host epithelial pathogen interaction because the lumen is not directly accessible to bacteria or virulence factors. Additionally, secreted materials such as small molecules, proteins, or mucus cannot be easily sampled from the 3D culture for downstream analysis. To address these limitations, we present a modified protocol for converting 3D human enteroids or colonoids to a monolayer. Colonoid monolayers secrete a thick apical mucus layer, allowing for ex vivo studies of pathogen-mucus interaction. This method aims to provide a tractable model to study the earliest intestinal host-pathogen interactions upon infection.
Colonoid monolayers will allow for a new way to study mucus biology, which is important in host-pathogen interaction, microbiome studies, and inflammatory bowel disease. This method can be applied to other mucus-secreting organs, such as the upper GI tract, respiratory system, and female reproductive system. Visual demonstration will help the user with mucus preservation during monolayer growth and during fixation for immunostaining. Demonstrating the procedure will be Carl Declodny, a staff scientist from our laboratory.
After incubating the coated cell culture, aspirate the culture medium from the 24-well plate and replace with 1 milliliter ice-cold harvesting solution per well. Use a mini cell scraper to dislodge and break up the basement membrane matrix pellet, paying particular attention to any material near the well edges. Then agitate the plate on an orbital shaker at approximately 200 revolutions per minute and 4 degrees Celsius for 30 to 45 minutes. Use a P200